Welcome to G Fun Facts Online. Imagine if we had a library containing a book for every living thing that has ever existed. A library so vast, it held more information than the entire browsable internet. Now imagine that all the pages were torn out, shredded into tiny fragments, and thrown into a single enormous pile. This isn't a dystopian fantasy, it's the reality of modern genomics. Over the last 20 years, we've sequenced the DNA of countless organisms, creating a torrent of genetic data that now exceeds 100 petabytes. That's 100 million gigabytes. This digital library of life should be a goldmine, holding the secrets to curing diseases, understanding evolution, and engineering a better future. But there's a problem. The sheer volume and chaos of this data have become a barrier. It's like being thirsty in the middle of the ocean. The very abundance of information was making it impossible to use. Scientists wanting to find a specific genetic sequence had to download massive terabyte-sized datasets, a process that could take weeks and required supercomputers, only to find that their needle wasn't even in that particular haystack. The paradox was maddening. We had the code of life at our fingertips, but we couldn't read it. That is, until now. What if you could just Google it? Welcome to the story of MetaGraph, the search engine for DNA. Developed by a team at ETH Zurich, led by Professor Gunnar Ratch and Dr. Andre Kallas, MetaGraph is a revolutionary tool that does something that was previously thought to be impossible. It allows scientists to perform a direct, full-text search on the raw DNA, RNA, and protein sequences themselves. A researcher can now paste a genetic sequence into a search bar, and within minutes, find out where that sequence has appeared across millions of datasets, from microbes in the deep sea to human gut bacteria. This simple act eliminates the need for those massive downloads and complex analysis, fundamentally changing how we do science. So how did they tame this data beast? The genius of MetaGraph lies in its clever combination of data structures and algorithms. First, instead of treating genetic sequences as long, isolated strings of letters like A, C, T, and G, MetaGraph breaks them down into an interconnected network, or a graph. Think of it this way. Instead of storing millions of copies of the sentence "the quick brown fox," you store the overlapping pairs, the quick, brown fox. If another sequence is a quick brown dog, you don't need to store quick brown again. You just create a new link, from brown to dog. MetaGraph does this with DNA, breaking it into smaller, overlapping fragments called k-mers. Each unique fragment is a node in the graph. This elegantly collapses all the redundant information. A tiny genetic sequence that appears in thousands of different species is only stored once. This turns a disconnected pile of shreds into a connected web of life. But knowing a sequence exists isn't enough. You need context. This is the second innovation, annotating the graph. The MetaGraph team developed a way to attach metadata, like the species, the geographical location, or the sample type, directly to the nodes of the graph. It's like taking that web of word pairs and adding a label to each one, saying this pair came from Moby Dick, Chapter 3, or this one came from a tweet sent yesterday in Japan. This turns the graph from a simple collection of sequences into a rich, searchable database of biological context. Now, when a scientist searches for a sequence, MetaGraph not only finds it, but also tells them everything about its origins. The final piece of the puzzle is perhaps the most magical: compression. Using advanced mathematics and what are called succinct data structures, MetaGraph can compress these global genomic datasets by a factor of 300 to 380. This is like having a zip file that is hundreds of times smaller than the original, but you can still search inside it without fully decompressing it first. This means those 100 petabytes of raw data can be compressed into a few terabytes, a size that can fit on a handful of standard hard drives. A task that once required a supercomputer can now be done on a regular server, and the cost of a large query can be less than $1. It makes large-scale genomics accessible to almost everyone. This isn't just a theoretical improvement. It's already having a massive real-world impact. Consider the global crisis of antibiotic resistance. Understanding how resistance genes spread through bacteria is critical. The MetaGraph team took on the task of analyzing over 240,000 human gut microbiome samples from around the world. Using traditional methods, this analysis would have taken months, if not years. With MetaGraph, it took about an hour. Because it searches the raw, fragmented data directly, it found resistance genes even when they were just tiny pieces in unsequenced, unknown bacterial species. The analysis even revealed geographical patterns of resistance that matched up with known antibiotic usage in different countries, providing invaluable data for global health surveillance. The applications are truly mind-boggling. In an era of emerging diseases, MetaGraph can act as an early warning system, scanning environmental samples for the genetic signatures of new viruses or bacteria. In medicine, it can help researchers find rare genetic variants linked to disease, paving the way for personalized treatments. The world's microbes are a treasure trove of novel enzymes. A similar search platform recently discovered over 200 million natural versions of a plastic-degrading enzyme. MetaGraph can accelerate these kinds of discoveries, finding enzymes to create new biofuels or break down pollutants. For the end user, it's designed to be simple. There's a web interface where you can just paste a sequence. The results come back in minutes, with interactive maps showing where the sequence was found, charts, and even AI-generated summaries. Now, MetaGraph is a landmark, but it's important to place it in context. For decades, the workhorse of bioinformatics has been a tool called BLAST. But BLAST and MetaGraph are for different jobs. BLAST is like using a search bar in a curated library of published books. It's excellent for finding a known gene in a known genome. MetaGraph is different. It's like a content discovery engine that searches everything. Every published book, but also every rough draft, every scribbled note, and every torn page in the entire library. It searches the raw, messy data archives, finding things that aren't in any curated database. A more recent contemporary is a platform called Logan. Logan also aims to search these massive archives, but it takes a different approach. It first tries to assemble the shredded pages back into longer strips or contigs, and then indexes those. This is also incredibly powerful, as shown by its discovery of those plastic-degrading enzymes. You can think of them as complementary tools. Logan rebuilds the fragments first, while MetaGraph searches the fragments directly. Both are moving us toward the same goal of making all genomic data accessible. Of course, the road ahead has its challenges. The sheer amount of new data being generated every day means keeping the MetaGraph index up to date is a constant battle. The team is always working on better compression and faster algorithms. But perhaps the biggest question is an ethical one. The democratization of a Google for DNA raises concerns about privacy and genetic discrimination. As the tool becomes more powerful, could it be used to identify people with genetic predispositions to diseases without their consent? The scientific community is actively debating how to build ethical guardrails around this technology. One of the developers, Dr. Kalis, mused that like the early days of Google, we probably can't even imagine all the future applications. He envisions a future where you might use a handheld sequencer and a tool like MetaGraph to identify the plants on your balcony. It sounds like science fiction, but the pace of change in this field is astonishing. MetaGraph is more than just a clever piece of software. It's a paradigm shift. It's the key that unlocks the library of life, transforming a crushing deluge of data into a fountain of knowledge. By making the world's genomic information searchable, it's empowering scientists to ask new questions, uncover hidden connections, and accelerate discovery in ways we previously could only dream of. The breakthroughs of tomorrow might not come from a new experiment in a lab, but from a simple search query, unearthing a secret that was hiding in the data all along. The era of petabase-scale genomics is here, and with tools like MetaGraph as our guide, the possibilities for what we can discover are truly boundless. Thanks for listening to Fun Facts Online.